Here we go. Okay, so we talked about the central dogma last time. It's the flow of genetic information into a cell. So the DNA contains like the blueprint for everything. It is safely secured in the nucleus. We don't really want to take the DNA outside into the cytoplasm because it's not really safe. There are a bunch of enzymes in there. We don't want DNA exposed to that outside environment to keep it nice and safe and intact. So DNA is that first blueprint, and then it's transcribed in the same language, which means DNA to RNA is just nucleotide to nucleotide. So that's considered transcription. So it's transcribed into mRNA. mRNA is what's translated into proteins by ribosomes. So, for example, if you're applying for any, I don't know, government or like just anything, you're applying somewhere and they ask you for your official government issued ID and you need to submit this online. What you would do is submit a copy of it and not the real thing because you want to keep the real thing for yourself. You want to keep it safe. So that's the purpose of mRNA. mRNA is then shipped out. It's translated into proteins by ribosomes. So we're just going to look over that process. The transcription is a process in which the genetic information in a sequence of DNA nucleotides is copied into newly synthesized RNA molecules. RNA polymerase is the enzyme that transcribes DNA sequence into RNA molecules. This is the same RNA polymerase that's used in replication to create primers. So then we have these different types of RNAs. So mRNA is the one we all know. It's messenger RNA. It's the one we're copying DNA into. And it contains 3 base pair sequences called codons, which are complementary to the DNA base pair sequence. So the way we copy it is we have DNA. This is DNA. One strand is going to be template. One strand is coding. When this opens up, mRNA is transcribed by the template strand. So we use the template strand to get our mRNA. The coding strand will have the exact sequence as the mRNA because they're essentially they're both complementary to the template strand. So keep that in mind. These codons, or 3 base pair sequences, will specify specific amino acids during translation. tRNA, or transfer RNA, is the one that looks like this, kind of like a little T. It carries the amino acid on this side, and it has the anticodon right there. Folds into a 3-dimensional structure that acts as an adapter, meaning it converts the code to an amino acid, which is a whole different language. One end of the tRNA will bind to a specific amino acid over here, while the other end of the tRNA contains an anticodon, which will pair with the appropriate mRNA codon of the ribosome during translation. Next is the rRNA, or ribosomal RNA. It also folds into a three-dimensional structure. RNA and proteins form ribosomes that perform translation. So all of this, by the way, is created in the nucleolus within the nucleus. And then it's stripped out. So rRNA is just incorporated. It's a piece of nucleic acid that's incorporated into ribosomes. The three-dimensional RNA acts as a ribozyme, catalyzing the reactions needed in translation. So anything with zyme at the end is a catalyst or an enzyme. That's where we get zyme from. Okay. All right. To start transcription, the enzyme RNA polymerase must bind to a non-coding DNA sequence called a promoter. We went over this. Promoter does not code for any amino acids. Instead, serves as a binding site for RNA polymerase upstream from the start of the coding region of a gene. So we have the DNA, we have the start. The start codon is always methionine. And then everything after that is coding until the stop codon, which is where we stop translating. So upstream from the start codon, we have our promoter. Okay? So that's what that's saying. The promoter region has the TATA box because it's rich in thymine and adenine nucleotides. And transcription factors have to bind this to help RNA polymerase bind to the promoter sequence. The reason we need transcription factors is because we want to control gene expression. So if we didn't have any transcription factors, as soon as this DNA is exposed, RNA polymerase can come bind and then start producing all this mRNA that we might not need. So what we can do instead is say, okay, we need transcription factors. So when the transcription factors are present, that's the only time RNA polymerase can bind and translate or transcribe. And whenever we do need to express that gene, that's when we're going to express the transcription factors. Okay, let's see. So RNA polymerase, again, it's directional, 5' to 3'. So yeah, 5', we're going to have the phosphate end, sugar, and then base. So phosphate, sugar, base. This is what a nucleotide looks like. So this is the RNA strand. This side, this phosphate group, is attached to the 5' carbon on my sugar. So this is going to be the end, 5' end, and then this side is going to have a 3'. So the 3' carbon. The OH group, let me just I guess draw it like this. I don't want to confuse you guys. Oh, you're seeing that? Yeah, the OH group on the 3' carbon is exposed right here. So the next nucleotide that can be added is only going to be added to this right here. We cannot add any nucleotides to this phosphate. So the reason why it's directional is because the only group that we can add to is right here. So RNA polymerase will be stuck right here. So as soon as another nucleotide comes along, that looks like this. We're gonna take this and then stick it right here. And then so forth. And we're gonna keep growing. So that's what 5' to 3' means. So it's similar to how DNA polymerase adds new DNA nucleotides in the 5' to 3' direction. Yeah, so we went through this. The fact that it's anti-parallel, so the template strand will be running from, let's say it's 3', 5' there. The mRNA, I'll draw it in green, will be like 5' here, and we're growing in this direction, 3'. So it's anti-parallel. The sequence of base pairs in the RNA strand that's transcribed is complementary to the template strand. You guys need to know, you guys need to use this wording. Complementary is an important word. Use this whenever you're explaining stuff. Anti-parallel is another important word. You guys, the moment you guys start using these words, the person who's marking your test will be like, okay, this person knows what they're talking about. But you need to use it in the correct form, obviously. Okay, so they give you an example what complementary means. So if the template looks like this, the RNA sequence will look like this. The template strand is also known as the minus strand or the non-coding strand or the antisense strand. This is because the sequence of the mRNA strand that will be read by the ribosome during translation is actually not identical to the sequence of the DNA strand from which it was transcribed. As you can see here, this thing looks nothing like this thing because they're exactly the opposite. So that's why we call the opposite strand to the template strand, like the DNA strand that's opposite to the template strand, is called the coding strand because it'll have the same sequence as the mRNA, except that instead of uracil, it actually contains thymine. Okay. Remember Yeah, so this is the other thing we talked about with prokaryotes, is that since they don't have a nucleus, as soon as an mRNA is being produced, it's accessible by ribosomes. So while the mRNA is being copied or transcribed, we also have translation happening at the same time. What does that look like? So we have our, we have our DNA. I'm going to draw it kind of like a circle because bacterial DNA is circular, it's not linear. And let's say it opens up in a replication fork as such. And then we have our mRNA forming right here. Okay, as soon as my mRNA is being formed, I have ribosomes binding it and then translating that way. And as soon as this ribosome reaches here, I have another ribosome that can attach right here and then start going that way. And so you'll see a bunch of ribosomes attach as they start synthesizing protein. And the protein is going to be longer for the ribosome that's, farther down in their translation. And then the ones that just started translating are gonna have shorter pieces of protein like that. So the way you can know which ribosome has been stuck to this mRNA longer and has been translating longer, you can look at the piece of polypeptide that's being formed and look at the size compared. Okay, I hope that makes sense. You guys can stop me anytime and ask questions if something's not making sense. Okay, so most prokaryotic cells, mRNA transcript formed in transcription is immediately accessible to ribosomes and can be translated without delay. In eukaryotic cells, the initial mRNA transcript, referred to as pre-mRNA, so this is before the 5' GCAP is added, the poly A tail is added, before splicing occurs, it's called pre-mRNA. This needs to be modified before it can leave the nucleus of the cell and go to ribosomes. So 3 modifications we looked at. The first one is splicing out the introns and leaving the exons in. So structures called spliceosomes, which are made up of small nuclear RNAs, or snRNAs, and small nuclear ribonucleoproteins, or snRNPs, remove the introns from the pre-mRNA and then splice together the exons, the coding regions. These exons can be joined in different combinations. We looked at this last time. Different combinations to generate multiple RNA transcripts from the same gene. So essentially we can have multiple different proteins from the same gene. This is quite smart because instead of being limited to only one protein per gene, we can now have multiple possibilities and multiple uses of that single gene. So this alternative splicing, again, another term you guys should know, it's called alternative splicing, gives eukaryotes the ability to generate a greater variety of RNA transcripts from just one gene than what can be generated from one gene in prokaryotes. So prokaryotes don't do any of this. Keep in mind it's eukaryotes only. So this is the pre-mRNA up top. And now alternative splicing allows me to splice each intron out here and also splice out any exon that I might not need for that specific protein. So here you can see that if we take out all the introns and this last exon, what that's going to look like is, since we're removing from the right side, always from the right side, we cut the right side of exon, okay? Always. We cut from the right side of an exon. And then the right side of an intron. So technically it would be left side of a exon as well. So if I cut here and here, I remove everything between, between these two exons. If I cut right here and here, I remove everything here. If I cut right here and here, I remove everything there. So now I have 1, 2, 3. This is what my protein is going to look like. Now, if I were to cut from here to here, I removed all of that. And then here to here. Now I have exon 1, 3, and 4. This is what my protein ends up looking like. So this is the process of alternative splicing. You can also, for this last example, it's cut here to here. So all of that's removed. And now I have protein 3. Okay, so introns, while they're not coding, so they don't code for any protein like these, some of them may function in regulation of gene expression. Regulation of gene expression is basically whether I want to increase gene expression or decrease gene expression. So that's what regulation is. To protect the 5' end of pre-mRNA transcript from degradation before it can be translated, as I said, outside the nucleus, cytoplasm is not a fun place for not just nucleotides. Nucleotides are fine, but nucleic acids, so long chains of nucleotides, it's not a safe place. So what we need to do is protect it so that they don't degrade and start getting chopped off. So we put the 5' GTP cap on one end. GTP cap is guanine triphosphate. So we have a guanine, we have 3 phosphates attached, and then we're going to attach it to our mRNA, the 5' end of my mRNA. So where my phosphate group is exposed on the 5', that's where I add the G cap. And then the last modification is the 3' poly A tail, an enzyme, poly A polymerase, adds a string of adenine nucleotides to the 3' end of the mRNA transcript. So this prevents degradation of the transcript, whereas the 5' cap allows the shipment of my transcript. So nuclear pores recognize the 5' GTP and allow mRNAs with the cap to exit the nucleus. It also helps in initiation of translation when the RNA reaches the ribosome. The poly A tail, so remember we're building 5' to 3'. We're going to degrade this way as well. So 3' to 5' is how we degrade it. So this helps prevent degradation. So when the mRNA strand starts being chewed up from the 3' end, we basically have this cushioning of a whole bunch of adenines. So when it gets chewed up like that, we're just getting rid of these non-coding adenine sequences. That's fine. Until the whole mRNA starts getting like degraded, and that's when we'll need like new mRNA to be transcribed again. So mRNAs with longer poly A tails tend to have longer durations in the cytosol, which allows more copies of the protein to be generated. So if this is a protein, we want this protein for like a good chunk of time. We want to have a longer poly A tail so that the mRNA can last longer in that cytoplasmic environment. After the excision of the introns and splicing of the exons, as well as the additions of 5' cap and 3' poly A tail, the transcript is now referred to as a mature mRNA. So we went from pre-mRNA to mature mRNA. Now let's look at translation. Any questions so far? Okay. So translation occurs at the ribosomes. We talked about the different types of ribosomes present in prokaryotes versus eukaryotes. Ribosomes are found in cytoplasm in both prokaryotes and eukaryotes. Again, you guys have to differentiate the between these two. I would suggest you make a little Venn diagram or something like this and have prokaryotes, eukaryotes, and start listing out the differences and then the similarities between them when it comes to transcription and translation and like different topics that we've talked about as well. This really helps. And do it from memory so you can judge where you're at with your understanding. Okay, so ribosomes in the cytoplasm, they're also found on the rough ER of eukaryotes only. In eukaryote cytoplasmic ribosomes usually translate proteins that will stay inside the cell. So the ribosomes that are just floating around freely in the cytoplasm, those produce proteins that just stay in within the cell, whereas the ones that are on the rough ER generally produce proteins that are being shipped out, or they're intermembrane proteins, like integral proteins in the membrane, and so forth. Okay. Oops. So, this is again what we talked about earlier. In prokaryotes only, the DNA is transcribed to mRNA and then translated to polypeptides all at the same time. So this is like this thing is called a polyribosome or polysome. I hope this diagram makes sense to you guys. You should be able to like look at directionality. So let's actually do that right now. If you look at this diagram, the template strand of this DNA, tell me the directionality. Can someone tell me the directionality for this DNA strand. This side versus this side. Is it 3' or 5'? Which one? And what's the thought process here? Okay, maybe I'll ask this. Which direction is this RNA polymerase going? Is it moving to the right or to the left?
Right.
Why? Like, how can you tell?
Like, the polypeptide chains get longer and the RNA polymerase starts on the left. Wait, never mind.
Wait, shouldn't it be on the left, going towards the left? Because the ribosomes are like almost done translation, right?
Wait, I don't even know.
How do you know the ribosomes are done translation?
I actually don't know. I'm just like assuming that maybe.
Okay.
I actually don't know.
Okay. So you guys always need evidence for the claims. So here, if it's moving this way, let's say it's moving this way, okay? Let's say that's your assumption. It means all of them are moving this way, right? Because of directionality, RNA polymerase only goes in one direction, 5' to 3'. Or well, if we're thinking of the template strand, it's 3' to 5'. But in terms of mRNA, it's building on the 3' end. So this side of mRNA would be 5', this side 3'.
So is it going left or right?
So we're looking at the size of the mRNA right now.
Okay.
If it's been transcribing for longer, as in transcription for this specific mRNA, if it's started sooner, then at this given time point that we're looking at this image right now, then that strand has to be longer than a more fresh mRNA strand, right?
Yeah.
Okay.
Then?
So which mRNA is older or has been like transcribing for longer? Which mRNA?
I would assume the one on the left.
This one?
Yeah.
So the shorter one?
This way. Yeah.
So if mRNA is shorter, we just established that a longer mRNA means it's been transcribing for longer, correct?
Oh, that's right. It's right then.
Okay.
My bad.
So this one, older. So it's been transcribing for longer. If we're moving in this direction, it means my RNA polymerase started here and ended up here. Correct? So my mRNA strand, should it be longer here or here? On the left or on the right?
Right.
It should be longer here. It should be longer here.
Yeah.
If this is the starting point. How can it be long if this is the starting point and I have 0 mRNA?
Wait, I'm so confused right now.
Okay.
So it's the left.
Yeah, but you guys
why-
have to understand why and be able
that's
to
longer.
explain it.
The longer it is, the older it is.
Yeah, but
So if you just started and it's short, and then the long one's on the end.
If you guys look up a video, that would be the most helpful thing for you because you can see it visually as time goes. But let me just do a little quick drawing here. So I'm going to say time point equals 0 time point equals like 5, and then time point equals 10. So in the beginning, I have my DNA. RNA polymerase starts here. Okay. At time 0, I'm going to draw my mRNA as green. So at time 0, I have no mRNA because I haven't had time to create mRNA. Now next, you're going to see my RNA polymerase here. Let me label this so you guys can follow along. I'm going to have some chain of mRNA produced. Correct? And then finally, as this RNA polymerase is moving this direction, let's say this is the end, I have my mRNA growing even more because it's transcribed more. Does this make sense?
Yes.
Okay, so now let's assume at time point 5, I have another RNA polymerase comes and attaches. There's available space. It comes, attaches. It's directional, so they only move this way. So now at time 5, for this RNA polymerase, I have no mRNA produced because that's the starting point. At time 10, I'll have this mRNA. It's moved along. It's done some transcription. So now I have some mRNA produced. Right? And now again, space is available here for another RNA polymerase to come and join and start moving that direction. As you can see, once it starts producing like mRNA, it's small. And then as it's moving in this direction, producing more and more, right? Synthesizing more and more, transcribing more and more, we're going to grow this mRNA. And then by the time it's down here, it's going to be a much longer mRNA. Make sense?
Yes.
Okay. So this, which mRNA strand is older?
The bottom one?
So we have 3 mRNA strands here. This one, this tiny one. Which one is older?
Wait, I can't see it if you're drawing it. I can only see. Is the one at the bottom?
Is it the longest one?
Yeah.
Yeah, this one.
Oh.
Longest one is always oldest. Why is this important? Because if I did not give you this directionality, okay? In fact, I didn't give you the time points, and I did not give you any of these previous drawings. I just showed you a snapshot of this. You guys have to know that the RNA polymerase is moving this way. It's not moving this way, clearly. Because if it moved this way, this strand should be much longer than this strand. Does this make sense? If it doesn't, I'm going to try and explain a different way.
It makes sense.
Yeah? Okay. For all of you, I hope. So let's go down here. Again, I have my RNA strand, or DNA strand down here. I attach an RNA polymerase somewhere, and it moves in the specified direction. So it always builds mRNA 5' to 3'. So again, you guys have to know that this mRNA strand is 5 prime here. Why is it 5 prime there? Can someone tell me? Why does this have to be 3 prime?
Because that's the beginning of the RNA polymers.
That's the beginning of the RNA polymerase?
No, like that's where the thing starts growing.
Exactly.
Over time.
So RNA polymerase can only add to the 3 prime. So that's where our RNA polymerase is adding right here. Okay. Now, which direction is RNA polymerase moving in this scenario?
Right.
Yeah, right, because this mRNA is larger than this one. The smaller mRNA is a more fresh mRNA. And so basically by the time this ribosome gets here, that's when this mRNA is going to grow to this size. And that's how we know this thing is moving this way. Okay. So if it's moving this way, RNA polymerase is moving that way. What is the directionality of my DNA?
Right.
No, like which side is 3 prime, which side is 5 prime?
Wait. Left is 3, right is 5, right?
Like that?
Mm-hmm.
That's right. So basically it moves in the opposite direction of which it's transcribing and translating. Or sorry, not translating, transcribing. Okay. So there you go.
But I don't get it. Why is there RNA polymers? Isn't that for like making mRNA? Why is it here?
Yeah, we're talking about transcription right now. Where else would it be?
Oh, okay.
Or, it's 12:30. And we're halfway. Let's take a like 8-minute break until 12:40. I'll see you back here.
guys? Someone just sum me up. Like, are you guys meeting today? I thought we were meeting on January 10th.
Okay, guys. Back to here.
I'm back.
All right. Does anyone have any more questions or concerns about this image?
No.
No.
Okay. So I'm going to move on. So you'll only see this in what kind of organism?
Eukaryotic. No? Isn't it prokaryotic?
Yes. Why?
Oh, shoot.
Why is it only prokaryotic?
Because there's like a bunch of ribosomes on it.
No, like, why is this possible in a prokaryote?
There's no nucleus, right?
Exactly. Okay, so all of this is happening in the cytoplasm. So that's why the ribosomes are present here, because it's the cytoplasm. Okay. Process of translation requires energy, involves 3 main steps in both prokaryotes and eukaryotes. There's initiation, elongation, and termination. So let's look at these. Number 1, initiation. The genetic code in mRNA. So we're talking about, again, to orient yourselves. This is now translation. We're talking about translation. First step, initiation. The genetic code in mRNA is read in 3 base pair units called codons. The start codon is AUG. What does this code for? What does AUG code for? Anyone know? It's an amino acid.
Methionine.
Methionine, yes. A tRNA with the complementary anticodon. In this example, the anticodon on the tRNA is going to be UAC. Okay, so this is my mRNA. This is my AUG. This is my tRNA with UAC. So that's what this is talking about. This is the anticodon. What's on the mRNA is called the codon. UAC tRNA will have a methionine attached. So that's why it's always the AUG, the start codon, is always a methionine. So the tRNA brings the appropriate amino acid to the ribosome, and the anticodon on tRNA pairs with the codon on the mRNA. This way, tRNA functions as an adapter molecule, linking the correct amino acid with the correct codon. This process is called initiation. Let's look at figure 16.3, which is initiation. You see the mRNA, it has the start sequence AUG. This is where translation begins. A tRNA carrying methionine with the anticodon that's complementary to AUG pairs with the mRNA, and then that's when our first amino acid initiates. Okay, so keep in mind the redundancy in the code, each codon on mRNA codes for one amino acid. So basically this mRNA sequence, there is no redundancy here. Like this codes for one amino acid, this codes for one amino acid. It's not like either lysine or isoleucine can bind to GGG. It can only bind one amino acid. However, the opposite isn't true. So multiple codons can code for the same amino acid. So GGG and GGC, so if we had GGC, it could code for the same amino acid. So that's what the redundancy in the code is. For example, the codons UCA, UCC, UCG, and UCU all code for the amino acid serine. This gives redundancy to the genetic code. This redundancy in the code may result in silent mutations, which do not affect the amino acid sequence of the polypeptide chain. So what's a silent sequence? It's when, for example, silent mutation. For example, you have UCA in your mRNA, or in your DNA, you have the complementary to that. Let's say this third nucleotide in the codon is mutated to C. So now you're going to have UCC. Well, it codes for the same amino acid, so this mutation makes absolutely no difference in the outcome or in the phenotype. So this is called a silent mutation. It is a mutation, it's still considered a mutation. However, the phenotype or the resulting amino acid is not affected, and so it's called silent. Almost all organisms on Earth use the same genetic code, which means basically UCA codes for serine in all organisms. This is evidence for the common ancestry of all organisms. So if we use a different genetic code, that would probably tell us that we don't have the same ancestry because the code isn't the same. So here, this is your amino acid table. You have the codons and the amino acids that they code for. You just have to learn the first letter. You look over here, which one's the first letter, and then the second letter, and then pick the third letter.
Wait, we don't have to remember.
No.
Okay.
Definitely not. You do not need to memorize any codons. The second phase, second step here is elongation. So after the first amino acid is placed in the ribosome by tRNA, the first one is always methionine. The ribosome translocates or moves to the next codon. So it's the ribosome that's moving. It shifts over here. So over here, as you can see, this is the center of my ribosome. I'm cutting it right down the middle. This is right down the middle. Now, if I were to just scroll down, again, this is the center of my ribosome. I am now aligned with the next codon. I want to be able to read my next codon. So I already read this one. I brought my tRNA in. Now it's time for my next one. And this thing is going to keep going. So the ribosome keeps moving to that side so that it can read each codon. So now that it moves, my tRNA carrying the methionine is now in a different spot in the ribosome, and my new tRNA that carries the amino acid for this codon is now in the center of my ribosome. And now a peptide bond between them is formed. Okay, a new tRNA with the appropriate anticodon and amino acid pairs with this codon. The ribosome then catalyzes the formation of a peptide bond, forming the beginning of a polypeptide chain. So once the peptide bond is formed between the amino acids, the first tRNA releases. So basically, once this bond is formed, this bond is broken. And now this tRNA, it does not carry any amino acids, so it's released. So now in your growing chain of amino acids, at the very tip of my mRNA, I'm going to see a methionine. And this is going to be, the methionine. Which side of the methionine is exposed? Is it the 5' or the 3'? Sorry. I'm out for the mRNA. So this, when we're moving this way, with the ribosome, we have the mRNA. We're moving this way because this is AUG. That's the start. Everything else we're going to copy is going to be on the right side of the start. The thing coding methionine is the AUG. So this side is going to be my 5'. This is my 3'. So even the ribosome is moving in the 3' direction. And then when it gets to the amino acids, it's also directional. So amino acids, on one side we have the NH2 or NH3 side, and then we're going to have the rest of the amino acids, and on this side we're going to have a carboxylic acid side that looks like this. So this is also directional. We say this is the amino terminal. This is the carboxylic acid terminal of the amino acid sequence. Okay, first tRNA releases its amino acid, and the first tRNA is released from the ribosome. This translocation of the ribosome and the addition of new amino acids to the polypeptide chain is called elongation. It's repeated one codon at a time until a stop codon is reached. So we will elongate, meaning we're gonna grow this polypeptide chain until we reach the stop codon. Termination. Stop codons, also known as nonsense codons, do not code for any amino acid. So over here, you have a bunch of stop codons here, 3 of them. You see any of these in a sequence, you know it's a stop. These release factors, so when the ribosome reaches a stop codon, proteins called release factors bind to the ribosome. So where normally you'd have a tRNA coming in with an amino acid, instead you're going to have a release factor coming in, and then it disassembles the polypeptide chain and ends translation, and the ribosome also kind of falls apart. So there you go. This is a stop codon, release factor comes in, and then this mRNA pops out. Small subunit and large subunit disassociate. Okay. Flow of information. I don't know if this is. So, this is already stuff you guys should know, so I'm not going to read it in detail. But basically, looking at the flow of information, not just information, but like end products as well. This is what it looks like. So, we have the DNA transcribe to mRNA, translate to protein, our ribosomes on the ref ER or within the cytoplasm, and then protein goes to the Golgi body. Then protein is in a vesicle, protein is outside of the cell membrane. So visually, this is what it looks like. DNA in the nucleus, we form pre-mRNA. Then we mature that mRNA so we can ship it out. And then the ribosomes on the rough ER will translate this mRNA. And then it's the protein right there as triangles. Goes into Golgi for further like snipping and editing. And then once it's ready, it gets shipped off to the cell membrane in a vesicle. And then we can just release that protein. Or if it's a membrane-bound protein, so it's like stuck in the membrane like that, then what you would see is, for example, this is my vesicle. This is going to be my protein on my vesicle. And it gets shipped out. So in this scenario, it would be like a protein right there. It integrates with the rest of the membrane, and so it ends up looking just like a protein spanning the membrane. So if you ever see a protein that looks like it's on the membrane of a vesicle or something, you know that it's going to be a cell membrane protein. Some viruses contain RNA as their primary carrier of genetic information. These are called retroviruses. Contain the enzyme called reverse transcriptase. The reverse transcriptase makes a DNA copy of RNA genome of the virus. So basically, we know how to go, like our bodies can go from DNA to RNA, but we can never go from RNA to DNA. We don't have the enzyme to produce DNA from RNA. But these viruses, they contain that enzyme. It's called reverse transcriptase. So we can go from RNA back to DNA. So what they do is they just spit out their own RNA. And our bodies can't go from like RNA to DNA. But since this virus also provides the enzyme, reverse transcriptase, we can take that RNA and produce DNA with it. With our own, like the nucleotides in our own cells are used to create this DNA. And then this DNA is then put into the genome of our cells, or whatever the host cell or organism is. Like it could be a dog, it could be a monkey. And then the host cell will then transcribe to mRNA and then protein. Just like that. So this is the infection process of a retrovirus. Yeah. And then they also tell you reverse transcriptase is less accurate than RNA polymerase. So retroviruses have a relatively high mutation rate. So this process is relatively less accurate, and so there's going to be a lot of mutations even going back to